This is AP Biology Final 6.3 to 6.7 by Alan Duenas. Up first is transcription and RNA processing, which is 6.3. So as you can see, there are three images, with the first one being DNA and the second one being RNA and the last one protein. And genetic material all pass through these. Genetic material stored in DNA molecules. So while DNA molecules are storing genetic material, RNA molecules are being used to facilitate protein synthesis using the DNA information. Protein synthesis is when we get instructions on how to make proteins from DNA information and transport them to the ribosomes so that the ribosomes can join amino acids in a certain order to make a protein. In order for protein synthesis to occur, several materials must be present. One is a supply of the 20 amino acids which make up most proteins. There are also three types of RNA that are required for protein synthesis to happen. The first one is rRNA, otherwise known as ribosomal RNA, which is used to manufacture ribosomes. The second one is tRNA, or transfer RNA, which carries the amino acids to the ribosome for protein synthesis. The last RNA needed is mRNA, otherwise known as a messenger RNA, and its purpose is to receive the genetic code from the DNA and carry it to the cytoplasm because that is where the protein synthesis is taking place since the ribosomes are located there. There are two steps into protein synthesis, and that is translation and transcription. The first one is transcription, and what it basically does is copy a gene sequence to make a RNA molecule. Then there is translation, which is when the genetic code gets translated to an amino acid sequence in a protein. Transcription is the first step to protein synthesis, and what it does is synthesize a strand of mRNA using the genetic code of DNA. RNA polymerase then binds to an area of DNA molecule in the double helix. After that, the enzyme moves along the DNA strand and selects complementary bases from available nucleotides and positions them in an mRNA molecule. The poly A tail has 100 to 200 adenine nucleotides, increasing the stability and helps from exporting from the nucleus. GTP cap is modified guanine nucleotide, and it protects the transcript and helps the ribosome attach to the mRNA. Pre-mRNA molecules have sections that are removed from the molecule called introns and sections that are linked or together to make a final mRNA called exons. Alternative splicing allows more than one protein to be made from one gene. The spliceosome removes introns and the remaining exons are ligated, which means to join together to form mRNA. Exons are the segments of RNA used to make mRNA, while introns are often junk. Translation 6.4. The process to make a polypeptide starts off with the arrival of the mRNA molecule at the ribosomes. While mRNA was being synthesized, tRNA molecules were come together with their specific amino acids according to the activity of specific enzymes. The tRNA molecules then began transporting their amino acids to the ribosomes to meet the mRNA molecule. The first step of translation is initiation, which makes ingredients come together to form the initiation complex, which is the molecular setup needed to make a protein. The ingredients needed are a ribosome, an mRNA with instructions for the protein will build an initiator, tRNA carrying the first amino acid in the protein. Regulation of gene expression 6.5. Gene expression, process by which instructions in the DNA are transcribed and translated into a functional protein by flowing through RNA as shown in the image at the right. Different types of chemical reactions regulate gene expression. Gene expression is a tightly regulated process that allows a cell to respond to its changing environment. It acts as an on and off switch to control when proteins are made. Key takeaways from gene expression. Gene expression is regulated by interactions between regulatory proteins and regulatory sequences, due to the presence of certain transcription factors or due to the modifications of DNA or histones. Epigenetics are reversible modifications of DNA or histones. The phenotype of the cell or organism is determined by the combination of genes that are expressed. Operons and operators. Operons are closely linked genes that produce a single mRNA molecule during transcription. Operators a sequence of that either inhibits or promotes transcription by binding to regulatory proteins. Gene expression and cell specialization. 6.6. What are promoters? Promoters are DNA sequences upstream of the transcription start site where RNA polymerase and transcription factors bind to initiate transcription. What initiates transcription? The interaction between regulatory proteins and regulatory genes and transcription factors initiate transcription. What determines the phenotype of the organism? The combination of genes expressed determine it. What blocks transcription and why? Negative regulatory molecules block transcription to inhibit gene expression. What is blocked from binding to the promoter? RNA polymerase is blocked from binding to the promoter. Small RNA fragments can have roles in gene expression. They are encoded by DNA and can break down mRNA in the cytoplasm. Most importantly, they can block transcription by binding to the mRNA or breaking it down. Mutation 6.7. Gene mutations are changes in the nucleotide sequences of a particular gene. A DNA nucleotide can sometimes be accidentally substituted for another nucleotide. Neutral, which means we got the intended protein. A positive, which means we got change benefited the organism. A negative, which means the change harms the organism. Alterations in a DNA sequence. Changes in genotype can result in changes in the phenotype through base insertions and deletions. Substitutions, deletions, and insertions are gene mutations. DNA nucleotides can either be removed or inserted into a sequence. Deletion and insertion change the order of the gene sequence. Can either lead to more or less production of protein. Changes in genotype result in phenotype. Disruptions in genes and gene products cause new phenotypes. What can cause random mutations? Errors in DNA replication, errors in DNA repair, radiation, and reactive chemicals. Genetics are the primary source of genetic variation. Changes in the genotype result in phenotype part 2. Also, errors in mitosis and meiosis can result in changes of phenotype. Errors in mitosis can change the number of chromosomes in an organism. This increases many things like disorders in humans or vigor in plants. It truly is up to the environment to determine if a mutation is detrimental, which means harmful, or beneficial, or neutral. Changes in genotype result in phenotype that are subject to natural selection. Natural selection, the process in which organisms better adapted to a particular environment have a higher chance of survival and reproduction, thereby passing on those adaptations to the next generation. Genetic changes that enhance survival and reproduction can be selected for and by the environmental conditions. Horizontal transfer of genetic information, increased variation, refers to the exchange of genetic information between different genomes or unrelated organisms. It primarily occurs in prokaryotics, and it contributes to the genetic variation in prokaryotic population. It includes transformation, conjugation, and transposition. Genetic variation is increased through sexual reproduction, independent assortment of the chromosomes, and crossing over. Transformation, uptake of naked DNA. Naked DNA is different by normal DNA by naked DNA not being protected by proteins and other molecules, comes from an external environmental source. Transduction is the transmission of foreign DNA into a cell. When a viral genome integrates with a host genome, DNA is transferred from one bacterium to another by virus. Conjugation, cell-to-cell transfer of DNA. It allows small segments of DNA to move from one section to another. An extension connects the cells and allows transfer. Alright, well then guys, this is the end of my AP Bio final.